Hello everybody and welcome back. In the last class we talked about cell theory, the two different kinds of cells, prokaryotic cells and eukaryotic cells, and we looked at the differences between them. And then we went over a basic outline of the important structures in the eukaryotic cell. Today what I want to do is talk to you about three very important structures in the eukaryotic cell: the cell nucleus, the ribosome, and the endoplasmic reticulum. And while today you'll be learning mainly about the anatomy of these things, what we are aiming for is an understanding of how cellular processes require the interaction of these components. So let's get going. Okay, so the nucleus is the genetic library of the cell. I do not like to call it the control center of the cell because the nucleus doesn't actually do that much. It is more like the repository where all the information that is needed to build a cell and to get it to carry out its functions and remain alive is contained. So it's much more like a library than a command center. The command center is really decentralized, and the commands are carried out by two molecules that are able to carry out activities known as RNA and enzymes. The nucleus is almost always the most conspicuous organelle in the cell, and it's often centrally located, although not always. And what's amazing about it is it's surrounded by a double membrane. As we'll come to find out, that means two phospholipid bilayers. They actually enclose the nucleus and separate it from the cytoplasm. The nucleus is studded with pores, and they connect the inside of the nucleus with the solution that surrounds it, called the cytoplasm. The nucleus contains the DNA. The DNA occurs in chromosomes. And chromosomes are long linear strands of DNA, and in the eukaryotes surrounded by proteins. And each chromosome contains genes. And so what I'd like you to do is make a drawing of this idea, and I'll show you what that drawing might look like. It might look something like this, where you see here's our cell, and there's the cell nucleus, and inside it are the chromosomes. And you can see they're long linear strands of DNA. And this is actually a replicated chromosome. So this is actually two copies of the same chromosome. And so a single chromosome actually looks like a long strand of DNA. And along that strand of DNA are all these different genes that occur at various locations on the chromosome. So this is what I want you to make a drawing of, is this idea that the single chromosome has genes scattered along it. And by the way, they're not continuous, as we'll find out. We'll find out a lot more about chromosomes and genes in the next unit on DNA. But for now, let's get back to cell structure. As I said earlier, the DNA does nothing but store the genetic information. The genes are expressed as follows, and that's the word we use, gene expression. First, they are used to create a transcript, which is known as messenger RNA. Or they can be expressed as other kinds of RNA that we're going to learn about later. Then the mRNA moves from the nucleus to the cytoplasm, where it is used to create a protein. So this first process of creating mRNA from DNA is called transcription, and the process of using the mRNA to create a protein is called translation. So let's make a flow chart of that too, just so you really understand it. And here it is. So we have, you can not put this in your drawing. We have transcription. DNA gives mRNA blueprints for making a specific protein. And by the way, the DNA doesn't make the RNA, it's enzymes, these protein molecules that actually open the DNA up, read it, and make an mRNA transcript of the information contained in the DNA. Then the mRNA carries the blueprint out of the nucleus to the cytoplasm and finds a ribosome, which we're going to learn about next. The ribosome is the site of translation, where the information in the mRNA is read and turned into a protein, or more correctly speaking, a polypeptide, but for now we can call it a protein. So down here would be the word protein. So we have transcription, movement, which we could call translocation, and translation, which is the changing of the RNA code into a protein, which is a kind of molecule about which we will learn more later. So, in summary, DNA codes for proteins and RNA. Proteins are made by ribosomes in the cytoplasm. And there's one other region in the nucleus, and I'm about to show you this, called the nucleolus. It's a darker region within the nucleus, and this is where the components, or what I like to call the subunits of the ribosome, are made. And those components, which themselves are RNA, are made by transcription also. So the ribosome is made of RNA, and we'll talk about that more in a second. So now here are some pictures of this whole process. Here is an artist's diagram, and here is what we can actually see in an electron micrograph. And as you can see here, the double layer has been torn apart in this prep, right? In other words, here are the two layers, the double membrane of the nucleus, and here you can see it's been torn apart. And then inside is the DNA, or the genetic material, which is referred to as chromatin. And then there's that dark spot inside called the nucleolus. The nucleolus is where the ribosomal subunits are made and put together, and then they're going to be transported out in these black dots that you can see on the endoplasmic reticulum and on the nuclear envelope are ribosomes. Notice the double membrane. Notice that there are pores going through the double membrane. They look kind of like grommets, if you know what a grommet is. And that's called a nuclear pore, and you can see them in this electron micrograph. And then you can see that the internal space between the two membranes of the nuclear envelope are connected with the inside of this layered structure called the endoplasmic reticulum. And so here is a close-up of pore complexes. You can see that's what we can see. And this is an artist's rendition of them. Let's move on to ribosomes. Ribosomes are organelles or structures that are made of ribosomal RNA. That means that the genes for them are transcribed into an RNA, and that RNA is then processed in the nucleolus to make the subunits of the ribosome. Those are moved to the cytoplasm where they come together to form a ribosome. And there are only two subunits in the ribosome. The ribosomes carry out protein synthesis in two locations. One is in the cytoplasm, or the cytosol, the cytoplasmic solution. And they're called free ribosomes. And the other is on the endoplasmic reticulum. In other words, they're part of the structure of the endoplasmic reticulum, or the nuclear envelope. And they're called bound ribosomes. So if one ribosome is free in the cytoplasm and another ribosome is attached to a membrane, which one of them do you think sings the song, "Don't Fence Me In"? I'll give you a second to think about it. Got the answer? Neither. Ribosomes can't sing. Okay, here's what we can see when we look at a micrograph. Here are free ribosomes, and here are bound ribosomes attached to the membrane of the ER. So this is the ER right here, and here are the free ribosomes in the cytoplasm between, or floating around inside the endoplasmic reticulum. And here is a model of a ribosome, the two subunits. And I like to say that it looks like a brioche hamburger bun. It has a large subunit on top and a small subunit on the bottom. Just as an aside, free ribosomes tend to produce proteins that are used within the cell, and bound ribosomes tend to produce proteins that will be placed in membranous vesicles and then exported outside the cell or packaged into organelles within the cell. And that'll become more clear to you later as we move on. Finally, we want to talk about endoplasmic reticulum. But before we do that, what you need to know is all these membranous organelles inside the cell are part of a system, meaning they interact with each other. And we're going to talk about the endomembrane system. They are in dynamic flux, meaning they move about and the membranes can merge with one another. So imagine having the walls of your room being flexible enough so that they could expand and contract, or if you wanted to move one room to another place, you could, and it would just merge seamlessly. That's what the membrane is like. The components of the endomembrane system include the nuclear envelope, the endoplasmic reticulum, which we are going to discuss today, and then these other things which we will discuss in the next talk: the Golgi apparatus, the vesicles, the lysosomes, the vacuoles, and the cell membrane. These components are either continuous with each other, such as the nuclear envelope and the endoplasmic reticulum, or they are connected via transfer by vesicles. As we'll see, these membrane-bound vesicles can merge with other cell membranes. Let's now look at endoplasmic reticulum. The endoplasmic reticulum accounts for more than half of the total membrane in many eukaryotic cells. In other words, it's big. It is continuous with the nuclear envelope, as I showed you earlier, and thus the inside of the ER is separated from the cytoplasm. So the effect of the endoplasmic reticulum is to compartmentalize the cells. And the importance of compartmentalization is within compartments, you can have different chemical environments. And that probably turns out to be very important in the cell. There are two distinct types of endoplasmic reticulum. The smooth endoplasmic reticulum, which lacks ribosomes, and then the rough endoplasmic reticulum, which has ribosomes studding its surface. Here you can see an artist's rendition. Here, the smooth endoplasmic reticulum, which tends to look pipe-like, and then the layer-like rough endoplasmic reticulum. You can see the ribosomes that are attached to the membrane. And here's a micrograph of the same thing. The smooth endoplasmic reticulum has a variety of context-dependent functions. So depending on what kind of cell it is in, the smooth endoplasmic reticulum may take on different roles. So in some cells it synthesizes, which means makes, fats. In others it helps metabolize or break down carbohydrates. It's involved in the storage of calcium. And in some cells, it can detoxify poisons, and this is especially true in liver cells, which have a large amount of smooth ER. It's also important for the synthesis of hormones in the brain, and so this is why I say the function of the smooth ER is context dependent. The kind of cell determines how the smooth ER is going to function. The rough ER produces proteins on the bound ribosomes, and that is its primary function. And then these proteins are moved about the cell inside transport vesicles. The rough ER is the membrane factory of the cell. In other words, it produces new membrane. So if a cell needs to grow, the membrane isn't produced at the site of the membrane. It's produced at the rough ER and then transported out to the membrane in vesicles. So today we have seen the nucleus and that it is the information center, the genetic library of the cell. We have learned a little bit about how that information gets expressed in the processes of transcription and translation. Transcription occurs in the nucleus where a messenger RNA copy of a gene is made, and then it moves to a ribosome outside of the nucleus where that information is used to make a protein. Endoplasmic reticulum has two types, the rough ER, which is the site of protein synthesis, and also membrane synthesis, and the smooth ER, which has a bunch of context-dependent functions. That's it for today. After watching this, go outside, look at nature, and ask biological questions. And I'll see you soon.